Welcome back to the fourth instalment of the Bridging series, where together we're consolidating the essential GCSE biology needed to give you the best possible start to A-level biology. In this video, we're moving on to genetics and evolution. We'll recap the core GCSE ideas behind protein synthesis, mutation, meiosis, genetic diversity, natural selection, and biodiversity, while also introducing a few of the concepts you'll study in much greater depth during Year 12. As always, don't forget the free misconception guide and summary sheets linked in the description below. Remember, the aim of this channel isn't simply to teach biology, it's to help you recognise the mistakes that cost students marks and understand how to avoid them before they become habits. Let's begin by revisiting one of the most important ideas in the whole of biology: how genes influence our characteristics. You'll already know from GCSE that genes are sections of DNA and that they contain instructions for making proteins. Those proteins go on to determine the structure and function of cells and ultimately many of our characteristics. For example, one gene may code for a protein involved in making melanin, influencing eye colour, while another may code for an enzyme that catalyzes a particular chemical reaction inside a cell. Throughout biology, you'll see the same idea repeated over and over again. Genes code for proteins, and proteins determine phenotype. So you'll also remember from GCSE that not every cell in the body is the same, even though almost every cell contains exactly the same DNA. A nerve cell looks and functions very differently from a muscle cell or a red blood cell, not because they contain different genes, but because different genes are switched on and off in different cells as part of the specialisation process. This idea of gene expression becomes a major theme throughout A-level biology. For a gene to be expressed, the genetic information in DNA is copied into a molecule called messenger RNA, or mRNA, in a process known as transcription. Messenger RNA is a single-stranded nucleic acid and contains bases adenine, cytosine, and guanine, but uracil instead of thymine. And you will be expected at A-level to know the full names of the bases, whereas at GCSE you will have got away with just the first letters. The messenger RNA then leaves the nucleus and travels to a ribosome, where its sequence of bases is read in triplets and used to assemble amino acids into the correct order during translation to create a protein. At GCSE, it's often enough to know the sequence of these events, but at A-level, you'll revisit each stage in much greater detail. You'll learn exactly how RNA polymerase copies the genetic code during transcription, why only one strand of DNA acts as the template, how codons on messenger RNA are recognized by transfer RNA molecules, and how ribosomes assemble amino acids into a protein with remarkable accuracy. Now that we've reminded ourselves how genes are used to produce proteins, let's think about what happens when the genetic information changes. A mutation is a random change in the DNA base sequence. You'll already know from GCSE that mutations can occur spontaneously during DNA replication, or they can be caused by environmental factors known as mutagens, such as ionizing radiation or certain chemicals. Now, whether a mutation has an effect depends on where it occurs and what it changes. Some mutations occur in non coding regions of DNA and have no effect at all. Others may alter the amino acid sequence of a protein, potentially changing its shape and function. Sometimes this can be harmful, sometimes it can be beneficial, and very often it has little or no effect on the organism. Now, at GCSE, you will have been introduced to how different types of mutations affect the genetic code. Substitutions, where a base change has occurred that may or may not change the amino acid coded for, or insertions and deletions, which can alter the amino acids coded for from the point of mutation onwards. At A-level, you'll be introduced to chromosome mutations, which involve larger sections of DNA containing many genes. A common misconception worth correcting now is that all mutations are harmful. In reality, most mutations are either neutral or have no noticeable effect. Harmful mutations can reduce an organism's chances of survival and reproduction, while beneficial mutations are much rarer. However, those rare beneficial mutations are incredibly important because they provide the new alleles on which natural selection acts. In fact, mutation is the original source of all new genetic variation. Without mutations, no new alleles would arise within a population, evolution could not occur, and natural selection would have nothing to act upon. Genetic variation is increased even further during meiosis, producing genetically unique gametes and providing the raw material for evolution. You'll remember from GCSE that meiosis is the type of cell division used to produce gametes, sperm cells in males and egg cells in females. This is different to mitosis, the process of cell division which produces genetically identical body cells. Meiosis produces genetically different gametes. As well as the different roles of meiosis and mitosis, you should remember that mitosis produces two daughter cells in one division, the daughter cells being diploid with two copies of each chromosome. However, meiosis produces four haploid daughter cells from the parent cell dividing twice. As you begin year 12, try to think beyond simply memorizing the differences and ask yourself why each process produces different outcomes. One of the key purposes of meiosis is to halve the chromosome number. Human body cells contain 46 chromosomes arranged as 23 homologous pairs, making them diploid. During meiosis, the chromosome number is reduced by half, producing haploid gametes containing just 23 chromosomes. This is incredibly important because during fertilization, a sperm cell and an egg cell fuse together, restoring the diploid chromosome number in the fertilized egg or zygote. If meiosis did not halve the chromosome number, the number of chromosomes would double with every generation. During sexual reproduction, fertilization itself is random. Any sperm can potentially fertilize any egg, creating a huge number of possible genetic combinations. And this is why brothers and sisters from the same parents are genetically similar, but rarely genetically identical. So we've seen that mutation introduces new alleles into a population, while meiosis and sexual reproduction generate an enormous variety of genetic combinations of different alleles. The next question is what happens to that variation? And the answer is natural selection. You'll remember from GCSE that individuals within a population show variation. Some of this variation is genetic, while some is caused by environmental factors. If environmental conditions change, or if organisms face a selection pressure such as a disease, competition, or predation, some individuals may possess characteristics that give them a survival advantage. And those individuals are more likely to survive, reproduce, and pass on their advantageous alleles to their offspring. And if this occurs over many generations, those beneficial alleles become more frequent within the population, and this is causing the population to evolve its characteristics. Now, one of the most important things to remember is that individual organisms do not evolve, populations do. Evolution is a gradual change in the frequency of alleles within a population over many generations. Another misconception that's worth correcting now is that organisms evolve because they need to survive. Mutations occur randomly, long before the selection pressure appears. Natural selection simply favours individuals that already possess these advantageous alleles. A great example of this is antibiotic resistance. You'll probably remember carrying out the antibiotic disc practical at GCSE. In this investigation, paper discs soaked in different antibiotics are placed onto a bacterial lawn growing on agar. If an antibiotic is effective, it kills or inhibits the growth of bacteria around the disc, producing a clear zone of inhibition. The larger the zone of inhibition, the more effective the antibiotic is against that particular bacterium. If there is no zone of inhibition around the disc, the bacteria are resistant to the antibiotic. And one of the common errors in terminology when describing antibiotic resistance is describing bacteria as immune. Bacteria do not have an immune system, so we cannot describe them as immune. So let's imagine a bacterial population where a random mutation has given one bacterium resistance to a particular antibiotic. When the antibiotic is taken, this acts as an environmental selection pressure. So natural selection occurs and the non resistant bacteria are killed, but the resistant bacteria survives, especially if you don't finish the course of antibiotics. Surviving resistant bacteria reproduce rapidly by binary fission, passing the resistance allele to its offspring. Over many generations, bacteria with alleles for resistance become increasingly frequent in the population. Now remember, the antibiotic does not cause the bacteria to mutate or become resistant. The resistant bacteria were already present before the antibiotic was used. The antibiotic simply acts as the selection pressure, favouring the survival and reproduction of bacteria that already possess the advantageous allele as a result of random mutation. Now, explaining the evolution of characteristics should always try to reference these 5 points: genetic variation, environmental pressure, natural selection, inheritance of advantageous alleles, and evolution over time changing the allele frequency. I teach my students to use this acronym GENIE to ensure that explanations nail all of the points on the examiner's mark scheme. This works at GCSE and this also works at A level. So before we move on to our final section, let's just recap. Mutation creates variation. Meiosis and sexual reproduction increase variation. Natural selection acts on that variation. And evolution occurs as advantageous alleles become more common within the population over time. And we must talk about the alleles that are advantageous, not the genes. Organisms have the same genes that are of the same species, however, they have different alleles. So, over millions of years, these evolutionary changes have given rise to the incredible variety of life that we see on Earth today. And this brings us to our final topic: biodiversity. At GCSE, you'll have learned that biodiversity refers to the variety of life in a particular habitat, ecosystem, or across the entire planet. This includes the diversity of species, the genetic diversity within those species, and the variety of different ecosystems. High biodiversity is really important because stable ecosystems are generally more resilient to environmental change. They are better able to recover from disturbances, are more likely to contain organisms with useful genetic variation, and provide humans with valuable resources such as food, medicines, and raw materials. Unfortunately, biodiversity is being reduced by human activities, including habitat destruction, pollution, climate change, and overexploitation of resources. As you've already learnt, conservation plays an important role in protecting endangered species and maintaining healthy ecosystems for future generations. So, this conservation may take the form of zoos, conservation areas, regulations, habitat restoration, breeding programs, seed banks, or even education. At A level, the step up isn't really the types of conservation, but more the depth of explanations. And generally, you can link the mechanisms of all of the different types of conservation to providing habitats, shelter, food sources, and mates to increase the carrying capacity of the ecosystem. You'll probably remember using sampling techniques to investigate biodiversity. Now, sampling is a particularly important practical because not only is it a required practical, but sampling is one of the 12 subsections of apparatus and techniques listed for the practical certification. And remember, I said that typically for AQA, at least 50% of this list appears in each exam series. So let's make sure we recap it here. Now, since it's usually impossible to count every organism in a habitat, biologists estimate biodiversity by taking representative samples. For plants and other stationary organisms, this often involves using quadrats. Laying quadrats at randomly generated coordinates allow us to estimate the abundance or percentage cover of species within a defined area. And we do this by finding a mean frequency of a species for the quadrat sampled and multiplying it by the area of the field. Now, when investigating how organisms change across an environmental gradient, such as from a woodland edge into the centre of a forest, transects are used. This helps us identify patterns in species distribution and how environmental factors influence where organisms are more likely to survive and live. At GCSE, it's often enough to know when quadrats and transects are used. At A level, you'll develop these practical skills much further. You'll learn how to design reliable sampling strategies using random sampling and systematic sampling, evaluate sources of error, and use statistical tests and techniques to compare habitats objectively. Rather than simply describing biodiversity, you're going to begin to measure it for real. Now, when it comes to motile organisms, you will develop your understanding of the mark release recapture technique, where organisms are captured, marked with a non toxic mark, and then released back into the habitat. After a period of time to allow random mixing, another sample will be taken in the same way at a similar time of day or year to ensure standardization. Now, providing certain conditions have been met, the number of organisms that are already marked compared to unmarked can be used to calculate an estimate of the population size using an equation that at A level you must be able to recall. Do check out my video on must memorize maths equations for biology. There is no equation list and you will be tested on some of these. I am always amazed at how many students overlook these equations as part of their revision. Now, as part of your biodiversity topic, you will also revisit classification. You'll remember that living organisms are grouped according to shared characteristics, helping scientists identify organisms and understand the relationships between them. You'll probably remember from GCSE that organisms are grouped into increasingly specific categories called taxa or taxons. As you move down the hierarchy, organisms share more characteristics and are more closely related. The main taxonomic groups are domain, kingdom, phylum, class, order, family, genus, and species. A useful mnemonic you've probably come across is Daddy King Pigs can only fly going sideways, although you're welcome to make up your own. There are three domains: archaea, bacteria, and eukarya. Organisms in the domains archaea and bacteria are prokaryotes, while the domain eukarya contains all eukaryotic organisms, including animals, plants, fungi, and protists. At the other end of the classification system are the genus and species, which hopefully you recall determine a species binomial name. A species can be defined as a group of similar organisms which can reproduce to produce fertile offspring. This definition will be central to understanding how new speciation events occur during evolution. Now, modern classification reflects evolutionary relationships, showing how closely related different organisms are, and these speciation events can be identified on phylogenetic trees, where lines meet to show when organisms' last common ancestor existed before speciation occurred. You'll remember that development of microscopes and finding of more fossils that fill gaps in the fossil record led to scientists changing their theories about species relatedness. And at A level, you'll develop further the ability to evaluate evidence of comparing species' phylogenetic relationships and explore how developments in DNA sequencing and molecular biology have transformed our understanding and allowed scientists to construct more accurate classification systems and evolutionary trees than were possible using physical characteristics alone. So that brings us to the end of this final bridging series video. Across the 4 videos, we've refreshed the key GCSE biology that underpins every topic you'll study in Year 12. Instead of spending September trying to remember GCSE biology, you will be able to focus on developing the deeper explanations and problem-solving skills that A-level demands. Now, please remember, Year 12 is not a warm-up year. The concepts you learn in Year 12 underpin the processes that you will learn about in Year 13. So please make sure you reflect on any tricky topics and embed regular recall and exam questions into your revision. As always, don't forget to download the free summary sheets and misconception guides linked in the description below. If you found this series helpful, I'd love you to subscribe so that not only do you build on your understanding of biology, but also how to develop your application of it. After all, knowing the content is only part of success. Being able to articulate it in the way examiners are looking for is what turns good understanding into top marks. So feel free to explore my other videos on the channel and watch out for my common misconceptions by topic videos. Thank you so much for watching. I will see you again soon and the very best of luck with your start to year 12.